Hello, my name is Kaden Todjo. I'm a rising sophomore at the University of Michigan studying biomedical engineering, and I conducted my research in Chicago at UIC in the NABA lab for ECM research. And my project is building a resource to explore the extracellular matrix and analyzing its proteins in lung and kidney tissue. So first, the NABA lab investigates the function of the extracellular matrix in development, health, and illness with a specific emphasis on cancer. Their goal is to develop how the ECM contributes to pathophysiological processes so that they can harness it and develop novel diagnostic and therapeutic strategies, primarily by combining traditional molecular and developmental biology techniques with cutting-edge proteomics. They are also developing open access resources that look to support and facilitate ECM research by providing thorough methods, tools, and information to the scientific community under their Matrizome project. For those who aren't familiar with the extracellular matrix, it's a common misconception that tissues are just clumps of cells, when in reality, a substantial part of them is in extracellular space, governed by an intricate network of macromolecules. And this is all known as the extracellular matrix. It stabilizes the function of the tissues and organs while regulating the behavior of cells in contact with the ECM, specifically the shape, proliferation, and migration of these cells. And it's composed of a variety of polysaccharides and proteins that assemble into fibers. Like you can see in this image to the right, collagen fibers coming from the top down, which are mainly responsible for the structural component of the extracellular matrix, as well as integrins and fibronectin, which are responsible for the adhesive and connectivity properties, as well as proteoglycans as well, which are composed of carbohydrates, proteins, and polysaccharides, responsible for the hydration and elasticity of the ECM. And it's these specific proteins that take on the roles of the extracellular matrix. And these proteins defined, are part of a defined aspect of the matrix called the matrizome. In the human genome, there are 1,027 genes that encode for the specific proteins within the extracellular matrix, and this is known as the matrizome. And the matrizome has two specific subdivisions: the core matrizome, which is responsible for the structural components of the ECM, and the matrizome associated section, which is responsible for the regulation of the components of the ECM. And given the matrizome's importance to the ECM and the fundamental properties of our cells, being able to visualize these structures and their interactions could help us to understand how its dysfunction can lead to chronic illness, which brings me to my first project for this summer. My task was to use HuBMAP's 30 OMAPs to curate an ECM-specific list for the scientific community. OMAPs, otherwise known as organ mapping antibody panels, are a list of antibodies used for the multiplex spatial imaging of healthy human tissue in each organ. I curated this list by first locating and gathering the antibody target information for all 30 OMAPs by downloading their corresponding CSV files. I next used a Matrizome website tool developed by the NABA lab to see which antibodies were actually targeting ECM-specific proteins. Then I curated an ECM-specific OMAP master list using all the antibodies that target ECM proteins. This list had the OMAP IDs and the organs they were used to map, the version of the OMAPs, the protein identifier for which protein the antibody actually targets, and whether the target was a collagen or a proteoglycan, as well as their matrizome division, and other technical information about the antibody in use, specifically for the experiment. I then revised the list by cross-referencing any of the newest changes and deployed it to the Matrizome website. And then finally, I used the list to observe patterns and observations within the OMAPs, beginning with this graph, which illustrates how many individual protein targets on the curated list have either one antibody targeting them or more than one antibody targeting them. For a little bit of background, about a year ago in the lab, a technician began some of the work that I have done this summer with version 7 of the OMAPs. And now that version 9 has been added with 7 additional OMAPs going from 23 to 30 and a revised section of 1 through 23, I wanted to observe the growth of the OMAPs from version 7 to version 9. This graph I created to illustrate those changes shows an increase of 17 antibodies overall. T Three for the glycoproteins, two for the collagens, two for the proteoglycans, the ECM regulators, and the secreted factors, as well as 6 new ones for the ECM affiliated. However, these numbers don't accurately represent the nuance of the antibodies across these versions, which is why I created this graph that depicts the portion of the matrizome with validated antibodies according to the OMAPs. The ECM glycoproteins have 12 unique antibodies, the collagen 6, the proteoglycans 2, the ECM affiliated proteins 13, the ECM regulators 3, the secreted factors 7. And this is represented by the outer rim, which represents the actual OMAP coverage, and the inner rim, which represents the total matrizome. And while the work done to visualize the ECM is impressive, it's severely incomplete, only representing a small percentage of the ECM. This is because it's common to see new antibodies that at least target a protein in the ECM, but not an antibody that targets a new protein we weren't already able to visualize from the OMAP list, hence the need for nuance, which is why, in addition to the Novel Labs' focus on developing proteomic methods for deep matrizome profiling, they also develop tools regarding the matrizome for the scientific community to use, tools like this validated list of ECM antibodies. However, the lab focuses on two aspects, one being the proteomic methods and developing matrizome tools, and the other, understanding the roles of the novel ECM protein SNED1 in development, health, and breast cancer metastasis, which is the main focus of my second project. But first, SNED1 is a novel ECM protein that influences the metastatic dissemination of tumor cells to distant organs. And this was determined by Alexandra Nava in 2014 by injecting human breast cancer cells of high and low metastatic potential into mice's mammary fat pads for about 6.5 weeks, and then harvesting those tumors. After enriching the ECM proteins from those tumors, it was detected that there were 26 specific to low metastatic potential and 43 that were specific to high metastatic potential. And among these highly metastatic proteins was SNED1. After injecting the mice again with a control and knockdown version of SNED1, it was observed that while protein knockdown didn't inhibit primary tumor growth, the spread of the tumor to other locations decreased significantly. Here you can visualize the results of this experiment, the left being the control and the right being the knockdown. The green dots are fluorescently tagged tumors, showing a significant amount less in the knockdown. These results help to support the notion that SNED1 does play a role in the metastasis of cancer. However, there is still a lot of information unbeknownst to the scientific community, like what cells are the top expressors of SNED1 in healthy tissue? Are there any consistencies? Are there any high levels of expression in a healthy body? Does SNED1 expression differ by organ, race, or even gender? Although there are many more questions that still need to be answered considering the importance of SNED1, these are the questions I chose to focus on. With questions 2, 3, and 4 being segues to answer my main question, question 1, which was where is SNED1 primarily in the body of adult healthy tissue? To begin to answer these questions, I used HuBMAP's RNA sequencing data on SNED1 from the available data sets on HuBMAP's data portal, which is an open data platform that assists in the visualization of single cell data in healthy tissue. In these data sets are multiple visualization tools, some of the ones I used being two different types of scatter plots, the left showing a distribution of cell types, and the middle showing ranges of SNED1 expression by single cells, and the one on the right being a violin plot showing ranges of SNED1 expression by specific cell types as a whole. In my interrogation, I found trends like universally low SNED1 expression across all six of the data sets, which makes sense because SNED1 gives rise to the metastatic ability in tumor cells, right? So in healthy cells, low expression is most likely to be observed. Within the lung data sets, the first four on the list, SNED1 expression ranged from about 1.14% to 3.33%. And dataset 4 was unique in its highest, it was the highest overall expression of SNED1. And dataset 5 and 6 towards the bottom were the kidney datasets that were either 1.8% or 2.4% of cells actually expressing SNED1. And overall, I noticed a major cell type shift by organ for the expression of SNED1. The lungs had more SNED1 expression in immune and connective tissue cells like fibroblasts, endothelial cells, and T cells, while the kidney has more specialized epithelial subtypes that are linked to nephron segments and high local specialization. And although SNED1 expression is low, there wasn't a clear observation within these healthy tissues, which raises additional questions considering that RNA sequencing data was available for SNED1, but not the protein data, which inhibits the type of inferences I can make, given that a lot of proteins in the extracellular matrix are long-lived, meaning that the RNA sequencing of the protein and the actual protein abundance don't always correlate as closely as I would like, which is why I know that as HuBMAP grows, the availability of features to be integrated, these databases, and the ability to visualize the structures and the interactions within the extracellular matrix will improve, which will be crucial for the advancement of prognosis and treatment for chronic illnesses. This is why the NABA Lab continues to work with HuBMAP through their Matrizome project, developing tools for the scientific community, while they also continue to focus on projects involving SNED1, because currently almost nothing is really known about SNED1, even though a better understanding of the protein could offer new methodologies for the prognosis of cancer tumor metastasis, especially in breast cancer patients. And although my work is a small aspect of what the NABA Lab has done, I'm grateful to have grown from my first biomedical research experience and learned how to improve my attention to detail, my prioritization, my time management, and my data visualization, all while learning how to interpret and interrogate data sets, the importance and inner workings of the extracellular matrix, and that the best mentors not only push the lab, but are always there and eager to help you. Additionally, I'd like to thank my mentor, Alexandra Nava greatly, as well as every other member of the lab. They've all taught me a lot, especially in my data visualization and the quality assurance of my work. I'd also like to thank HuBMAP and the NIH, as well as my family and my friends, especially my mother, who reminded me of the application after I had totally forgotten. So thank you again if you're watching. And thank you again to the NABA Lab for having me and all that you've taught me and for every fun moment in the lab. And for the rest of you, I hope my presentation and the work the NABA Lab has done has given you a new appreciation for the extracellular matrix. Thank you for your time and attention.
Well, that was wonderful. And let's all be honest, moms are not thanked enough. So that was I liked that. That was a great ending to, I think,
Mhm.
all of us. And I think a lot of our collaborators can feel the same way that they are not appreciated enough. So we do appreciate that. And your presentation was really, really done very well. We'll add one question if anyone has a final question, and then we'll do some closing remarks and we'll all be free. I know we are just a couple minutes over, but if anyone has any questions for this, hopefully we can end strong. No. Okay.
I have,
Of
I have a quick question.
course.
Thank you, Kaden. That was really great. And I liked to see the evolution from the OMAPs and the translation to looking at the cellular data.
Yeah.
Uh, That was very nice. So what would you, as a new user to all these tools, what would your recommendations be to the researchers that are out there on best practices?
To the other researchers who might use these tools,
Yeah.
um, I'd say that most of the time it takes a lot of due diligence looking through it and a lot of discipline to keep going, regardless of your setbacks. Because when I was first given the project, it was assumed that I would be able to look at the protein data of SNED1 and not just the RNA sequencing data, but that turned out not to be true. So I had to do a little pivot to focus on the specific RNA sequencing, as well as talking to another member of my lab, Aman, who taught me the importance of looking at the difference between RNA sequencing and protein abundance and the fact that those two don't always correlate as closely as we would like, especially in the extracellular matrix.
Thank you.
It was wonderfully answered, actually. Very, you knew what you were talking about, so that's always good to
Mhm.
know. Again, thank you everyone